Dr. Greek?
Thank you. I agree with Eric in part about not dwelling too much on the past and looking at how things could have been done, but how they were done, and so on and so forth. I think that's largely an exercise in futility, except for the fact that if the animal model community is going to draw from the past to justify the present, then it is very legitimate to say, okay, this is how Harry Harlow did it, but did he have to do it this way? If you're going to maintain that the animal model is vital, then you have to go back in history and say, we could not have done it this way here, we could not have done it this way here, and so on and so on. Otherwise, you're saying that the animal model is sufficient. It's not necessary, but it's sufficient. And finally, my grandmother was raising children during the time that these memos from the government went out. She had a third-grade education. She lived in the middle of Michigan, and she thought that was just the stupidest thing she'd ever heard. Now, maybe that's because she lives from the Midwest and, had a little more common sense than other people. But if this was some great scientific mystery during that time frame, gosh, I don't know any of the people who were being hoodwinked by that.
Okay. Dr. Greek, question for you. Regarding Dr. Sangren's example of breast cancer gene, the probability of finding that same gene in humans by coincidence is astronomically small. In this case, would you agree that the rat model was predictive? And if so, are there some areas of research where animal models could be predictive, or would you say they are universally inappropriate?
I would say they're universally non-predictive because, again, if you're going to say that the rabbit is predictive for birth defects, for example, in thalidomide, you have to at least show 19 out of 20 times where the rabbit got it right. All right? In science, and in drug testing in particular, even 19 out of 20 is inadequate. Vioxx was pulled off the market because a very, very small number of people were harmed by it. Millions, and perhaps even as high as 1 billion, people were actually helped. All right? So in science, again, prediction has a very specific meaning. And with regard to the breast cancer example, yes, that's a very good question. Most of the gene association studies that we do today are not done in animals for reasons that I think should be obvious. They are done in humans, and they're called Genome-Wide association studies. And if you pick up the New England Journal just about any given week, you will find on the cover a Genome-Wide association study that has identified in humans what genes are associated with cancer, heart disease, and so on and so on. So you have to study humans. There was a study done about 10 years ago that identified 9 genes, I believe it was, that caused seizures in mice. And everybody went looking for those genes in humans, and they found them in people who had no seizure disorders whatsoever. So occasionally getting it right, and I'll just grant you that animal models occasionally get it right. That's not prediction.
Okay. Eric?
I think my response to that highlights the principal difference in the way we approached this presentation. I believe that I am using and continue to use prediction in a precisely appropriate way. Looking back historically, probably that's something that Ray and I should have talked about before, coming up with a definition. But in some ways, I think it's better this way because you can see 2 approaches. And again, as I likened that to the Zeno's paradox, the approach that scientists use is to make predictions that turn out to be accurate using animals. They do that based on their understanding of the animals, similarities and the differences from humans, and that mammary cancer, that breast cancer example I gave, I picked very deliberately because according to my understanding of the word prediction, that data from the rat predicted something in humans that had not been identified by a human genome-wide study. And that is how scientists work, over and over and over again. Every finding doesn't translate to humans, but that's part of the scientist's job, to identify those that do. Every seizure gene in a mouse may not cause seizures in humans. But there are some other cases. There are some storage diseases, for instance, where there are defective enzymes and you get accumulation of byproducts in cells in the nervous system. And there, the animals happen to be exceptionally predictive for humans. The diseases that develop are identical, at least to all of the tests that we can provide. So there are examples like that. The 9 times out of 10 or the 19 out of 20, that's sort of a straw dog, or in veterinary medicine we say a straw dog argument, a straw man argument. That's really irrelevant to the way scientists perform their work. We instead, as shown by this particular example, identify those cases where we can get information and then take advantage of that.
Okay, thank you. Dr. Stein, we'll continue with you here and with the next question. The thalidomide tragedy of the 1960s demonstrated quite well the necessity of animal testing. If more animals had been tested, wouldn't the thalidomide tragedy have been prevented?
If, in fact, the study had been done on the particular type of rabbit that showed the same response, then that would have been identified. I don't think, though, the direction that we want to take is to test every drug on every animal, just as the direction we don't want to take is to test every drug on every human being. The direction that we want to take is to identify those areas where humans and animals have similarities so that we can provide more information, so that we can gather more information, so that we know which are the appropriate animals to use. At the time, we did not have a way to do that. Now, our ability is improving as we learn more about animals and as we learn more about humans.
Dr. Greek, do you have a response?
Thalidomide is actually a very good example. If I can get my computer to work, I'll even show you a slide. This is from a book on teratogens, 1976. It says in approximately 10 strains of rats, 15 strains of mice, 11 breeds of rabbits, 2 breeds of dogs, 3 strains of hamsters, 8 species of primates, and other varied species such as cats, armadillos, guinea pigs, swine, ferret, in which thalidomide has been tested, teratogenic effects have been induced only occasionally. If as Eric suggests, we're going to pull every drug out of development that causes birth defects in one animal species, you're not going to have any drugs. Every drug on the market today will cause birth defects in some animal if given at the right time and in the right dose. That's called Carnovsky's Law. And this is from, again, a textbook in 1985. And it says the actual results of teratogenicity testing in primates, which have been the most disappointing in consideration that these animals use as a possible predictive model. And it goes on to say, of 15 listed putative human teratogens tested in non-human primates, only 8 were teratogenic in one or more species. And it's actually pretty much universally agreed upon if you read the textbooks of teratology. The example of the thalidomide disaster illustrates this problem particularly clearly. Such a medicine-caused disaster could no more be prevented with adequate certainty through animal experimentation today than it could have at the time. Look, you test on 10 different species, you get 10 different answers. Yeah, one of them is probably going to be what humans do. How do you know prospectively which species? Every drug we have on the market today will kill something. I'll guarantee you. If it kills one species or one strain of mouse, are you going to pull it? Are you not going to allow it through development? That's ridiculous. But that's what the animal model community would have you believe. They would say, well, see, the rabbit actually did predict this. No, I'm sorry, but the word prediction has a very definite meaning. And if you wonder what it is, ask your bookie, okay? If the bookie will allow you to bet, put a $10 bet on 6 different horses, okay, for only $10, I'd go with that bookie. But most likely he'll charge you $60, 10 for each horse.
Okay, thank you. Dr. Greek, if the NIH believes that animal models aren't predictive, why are they funding animal-based experiments?
The NIH at the meeting I attended was very specific in saying that the animal model for ADMET or ADMET testing was not predictive. Okay? And in fairness to the NIH by and large does not fund studies because they think they're predictive. The NIH is pretty outspoken for funding basic science research. All right. So if you would ask the NIH, do you expect this study to be a predictive model for humans, they'd probably look at you funny, because that's not the business that they're in. The reason NIH was at that meeting was because there was this very nice lady who got up at the very first of the meeting and said, I have a $100 million budget, and I want to give it to you because NIH is concerned that there is no predictive modality for drug testing, okay? And we are so concerned about this, we're going to give money to Pfizer, right, like they really need it, because we are so concerned about it. So I hope that makes sense.
Eric?
So the NIH funds a large variety of research, and part of the funding is, a large part of the funding is, in fact, there have been several recent initiatives in this area, goes specifically to animal models of human disease. That's how they classify it. So the NIH very clearly does believe that animals can serve as appropriate models for human disease. The data coming out from the laboratories that are supported shows that animals can serve as good models for human disease. And by that I mean predicting aspects of those disease. So that's why NIH continues to fund it, particularly now when funding of all federal agencies but one is very strapped. That funding continues for animals because they can see the benefits that have come from it.
Okay, Eric, here's another question for you. What are the sources of funding for animal research here at the UW, and what is the range of costs for animal studies undertaken here?
The sources of funding for animal research are varied. Most of it comes from National Institutes of Health. Some of it comes from the United States Department of Agriculture. Some of it comes from the National Science Institute. Much of it comes from private foundations. And what was the second part of that question? Not just the source.
What is the range of costs for animal studies?
It's really impossible to give a single number for the costs. That depends on the species that are involved and obviously the number of animals that are involved. The typical grant to an investigator from the National Institutes of Health is the R01 grant. And generally that's for approximately $200,000 a year and lasts for 4 or 5 years. That would be a typical budget for a laboratory that used animals, but also a lot of other supporting materials. It's not just the animals, but it's the types of analyses that are done. Some laboratories have more than one R01. I don't know what the average figure is for research dollars per laboratory on this campus, but I would guess it's somewhere in the $300,000 to $400,000 a year, with a very broad range going from zero up to several $1 million a year. I hope that addresses the question. It's the best I can do.
Dr. Greek, do you have any response to that?
I can add a little bit to that. This is from 1999. These are some of the costs associated with using animals. As you can see, it's a wide range. Mouse sales, again, this is from 1999. Mouse sales amounted to over $200 million in 1999. These are industry figures, by the way. Mice with a specific gene missing cost from $100 to $1,500. So we're talking about a lot of money that's being spent here.
I wish all that was coming to this university, but it's not.
It's going to some university. So it's a lot of money. Exactly how much money is it? The very few studies that have been done show that about 50% of the budget from NIH goes to animal-based research, okay? And that's from a GAO study in roughly 1985. I have the reference if anybody wants it.